Very good morning to the audience. At the outset, I wish to thank the organizers for giving me this opportunity to interact with you people here in the conference. Specifically, I am thankful to the Microbiology department, the faculty, specifically Dr. Sudha and others. And just I welcome some of my fellow colleagues, ICR colleagues. I could see here my esteemed chairman RSC for the farming system research and few other colleagues which I could see. So welcome you all here in very important session. And before my talk, just I wish to take a couple of minutes to introduce my institute here. Actually, I come from one institute named National Bureau of Agriculturally Important Microorganisms. This is the ICAR institute which deals with all sort of microbial genetic resources, its conservation, its maintenance and its application in agriculture. So the institute is located near Varanasi in Uttar Pradesh. It's 120 kilometers far from Varanasi. And we are the custodian of national wealth. This institute is unique of its own kind, even among the bureaus. Within ICR system, you might be knowing that there are 5 bureaus, deals with different commodities like plants, animals, insects, fishes, and 5th one is the microbes, which I represent. We are the only institute in Southeast Asia who are having the status of International Depository Authority for the agriculturally important microorganisms under Budapest Treaty by World Intellectual Property Organization. So that is the uniqueness. In addition to that, we are mapping the microbial diversity of the country, not only the rhizospheric microorganisms associated with different crops, as well as we are mapping the diversity from forest, from extreme regions like mangroves, like the psychrophilic conditions like hypersalt, hypersaline conditions, etc. Here now the topic given to me is the conservation, maintenance and utilization of agriculturally beneficial microorganisms, a holistic approach to the sustainable agriculture. So since audience is like heterogeneous, what I think that in addition to microbiologists, there are the workers, researchers, as well as practitioners and students of different disciplines. So I will try to just keep myself restricted for few of the general things only, just to sensitize you, just to have a glimpses of microbes which are important and what are they doing. The first speaker of this session has already narrated in a very beautiful manner that what the challenges we are facing specifically in agriculture. So you know that there will be the expected population of 9 to 10 billions by 2050, and we have the greater responsibility to feed all those populations, not only the human populations, but our livestock, our other animals, everything which is residing on earth are directly or indirectly dependent on the plants. So this is our responsibility of judicious use of our resources for a sustainable kind of agriculture, because in our country we know that because of the green revolution, that was the demand with the time that we had to feed our huge population and we had to become the independent food secured country. But as a side effect, or as the some of the associated effect of green agriculture was, we lost our crop diversification, we lost the health of our soil system because of the excessive use of fertilizers, because of the certain high yielding varieties, because of the plant breeding programs that favored a certain kind of development in favor of a crop. So crop diversification last, microbes also last with associated with those wild species as those underutilized crops in our country. So our soils are becoming day by day becoming the imbalanced kind of soil where the nutrient imbalance is there, where the microbiological ecosystem imbalance is there, and we presume, we hope, and we work for the restoration of those kind of soil system. And when we talk about all these things, I would like to say that the last revolution in agricultural yield was delivered by a combination of the genetic engineering, chemistry-based intervention, mechanization, and improved crop management. But what is the next? So next one may be very well come from influencing the microbial communities. Now the importance of microbial communities is being recognized by each and every sector, not only the agriculture, but the pharmaceutical industries, but some industrial interventions related to the agricultural byproduce. So now the importance has been well recognized. Now the time has come that how to channelize all those informations which we already have with us, as well as the informations which are coming with the advancement in our research programs. So multiple studies have supported that the soil microflora are responsible for greater crop yield and indicate that even the microbiome, the plant microbiome, plant microbiota, which already reside inside the crop, they are the major players for the crop health and their capability to cope with the biotic as well as abiotic stresses. Even when it comes to environment, I think that some of the environmental scientists is also here. So when it comes to environment, microorganisms are known to help to regulate the aberrations as a result of climate change. That is also there. So the present days science is facing dual challenges. One is the raising global food demand, as well as another one is the environmental impact. So we have to design our research, we have to design our strategy for the crop production, of course including sugarcane and means cash crops as well as other cereal pulses and other crops. In every sector we need a balance. However, there is ample scope in cash crops like sugarcane and others, because in sugarcane I think that microbiology is in a nascent stage. Very little work has been done on sugarcane microorganisms. However, in general, if you see the publications, because the research publications are good indicative of the things, of the work which is being done globally. And I tried to see it since 2019 till today, means in past 5 years there are more than 17,000 publications on this aspect. If you Google it, you will find that there are more than 17,000 results which shows the research publications. It is not the articles and other publications or newspaper articles or other things, but if you see the research publications, they deal with the biotic and abiotic stress management through microorganisms in the crops for better yield. And most of the publications are coming in good impact journals, having good citation and good coverage across the continents, across the countries. So this itself shows that our scientific fraternity are working in this direction. And if we think that what are the key factors for the sustainable agriculture, what should be our goal, how we can achieve the sustainable agriculture, our aim should be reduce the chemical inputs. We should use ecological strategies for the management of pests and diseases. Then nutrient cycling is another important thing to bring back the nutrients in soil, to bring back the soil fertility and soil health, and expanding the production to meet the need of growing population. So there are different ways and means for the sustainable agriculture, right from agroforestry, intercropping, crop rotation, manuring, then bioagents, conservation, tillage and green manuring. And most of these are directly or indirectly related with microorganisms. If you see what the microorganisms are doing, they are helping in soil processes, they are helping in ecosystem functioning, they are helping in food and nutritional securing pillars. They are the major players for nutrient cycling, you know, carbon and nutrient cycling. They are the contributors for even carbon sequestration and soil carbon in the soil, organic carbon in the soil. They are players for the bioremediation, degradation, immobilization and modification in the soil system. They are the players for other things to maintain the soil biota and soil formation as well as plant soil interaction, etc. So they are the, in nutshell, they are the players for availability of nutrients to the plant, its access, its utilization and its stability in the soil system. All are governed by the microorganisms, and this hidden world of microorganisms contribute towards more than 90% for the plant growth, either through the better nutrient availability or through altering the root architecture or providing the plant a kind of immunization against the abiotic stresses or providing the immunity against certain diseases, against minor pathogens. It was very well said by the first speaker that there are certain emerging diseases, like she took one example of Coletotrichum gliosporidis in flowers, but in our country, in India, the challenges are even more. Because I will cite one example, since I could not make my presentation in that direction. The earlier program was given to me where some of my colleagues from IRI were supposed to deliver the talks, like my colleague Dr. Archana Suman and Dr. Lavalin Shukla. So I purposefully skipped the presentation which was expected by them. So those slides are not here, but I wish to tell something about that also. But just I wish to bring it to your attention, your notice, that in our country some of the minor diseases which were considered as minor from our college days, like leaf spots in case of wheat, maize and other cereal crops, bipolaris species, Carvularia, etc. They were supposed to be the saprophyte and minor diseases in case of these cereal crops, but now they are becoming the major disease and becoming the part of the leaf blight complex, which was supposed to be caused by some major pathogen. But now we have published from our institute 2 to 3 first report regarding those pathogens. Similarly, we have seen that blast in Bangladesh, wheat blast, that was supposed to be the disease of rice by Pericularia oryzae. That is the major problem in case of wheat, and we should, however, we should manage in our country, and that disease is not here. Similarly, a few other diseases are also there which used to be considered as minor but becoming major. Why? Because of the imbalanced use of fertilizers as well as the breeding resistance breeding program favored certain gene pyramiding that gave an opportunity to those minor pathogens to become major, because we could not take on board that aspect in our breeding program. So just I will go very quickly because the time is less and I need not to narrate these things, but you know that the biological nitrogen fixation is one of the important aspect even for sugarcane crop also, and I will show you a couple of this published articles also on this aspect. But just I wish to highlight that 200 million tons of nitrogen is estimated to be fixed annually by the nitrogen fixing microorganisms, and you are better than me in calculating the economics of this thing, because these microbes, they don't take anything from you and they fix such a huge quantity of nitrogen. If you try to produce it chemically, it will be a Herculean task in terms of finances as well as in terms of the capacity building of the industries. So all those cyanobacteria, azotobacter, clostridium, some azola like cyanobacteria association with pteridophytes, cyanobacteria association with gymnosperms and bryophytes, etc. All those fix atmospheric nitrogen. Then you know that nodulating microorganisms in legume crops also contribute a lot. Same thing is true for the phosphorus solubilization. Consistently we are adding the phosphorus fertilizers, inorganic fertilizers in soil system, and we know that 80% of those fertilizers become non-available to the crops because of its inherent chemistry to bind with the soil particles. So what the microorganisms do, they solubilize those phosphorus which is already available in the system and make it available to the plant roots, including mycorrhiza and other some of the bacteria and fungi. They contribute a lot for this kind of activities. So PSBs are the major players for the phosphate solubilization, and I request to stop or to minimize using these fertilizers, because our country is paying a huge amount on these fertilizers, and use the microbiologicals developed by our institute or the institutes under ICR system. There are several institutes who are working on it. So the same thing is true for potash, same thing is true for zinc. For potash also, we know that the structural potassium is not available. It is available in the form of mica, feldspar, biotite, etc. So these are non-exchangeable potassium is being mineralized by the microorganisms and make it available to the plants. Zinc, now the scientific community has realized the importance of zinc, and in addition to giving the elemental zinc, now microbes are there that can give you a better uptake of zinc as well as iron and to the crop plants, and that can be utilized to meet the demand. So this I wish to just wanted to show you from our institute this work we have done in wheat and some other crops. We have shown the localization of even those endophytic microorganisms which are associated with crops, and they contribute very much for the zinc and iron fortification, and even they increase the availability of iron and zinc in seeds, sweet seeds. So a proper sink is there. Biofortified crops can be developed using these kind of microorganisms. Then they also work for the hormone production, IAA, gibberellic acid, etc. We know since ages, but certain volatile organic compounds produced by these microorganisms also take a very important role. Then certain signaling molecules like AHLs, etc. also plays very important role in the plant well-being, crop well-being. Other attributes are like the biopesticides I told, then the remediation from heavy metal like arsenic and cadmium also. The plant can provide certain kind of protection to roots and to reduce the uptake of these harmful heavy metals in our crops, which ultimately leads to our export increase. Because if you remember 6 to 7 years back, Iran has just refused our basmati rice because of the traces of some of the heavy metals like arsenic and other things. Biocomposting is another thing. Your sugar industry and other industries are also trying to promote the composting and fortification with trichoderma. I will discuss later on if time will permit. Then just I wanted to sensitize you about certain novel mechanisms which might not be known to most of you, that these microorganisms alters the root architecture and they improve the rooting of these plants for better absorption, and when root area, root volume, and this branches in roots, when it is enhanced, it gives you a better absorption for these things. So in nutshell, this is the mechanism for biotic and abiotic stress amelioration. This is little technical, so I will skip this slide here, because it will take much time if I will try to explain all those mechanisms, but they contribute, they induce the systemic resistance, they induce the defense mechanism in the plant, they mitigate the abiotic stresses for against the various kind of challenges the crop is facing. Now, since this meeting, this international conference is on sugarcane, I wish to bring to your notice about a recent publication from my own research group, one of my student who is working currently in China in BMC plant biology. They have done a very good work in China, and I wish that this kind of work should be done in our country, because we are also a major grower for the sugarcane. Diversity of nitrogen fixing rhizobacteria associated with sugarcane they have encountered. They did a comprehensive study of plant microbe interaction for growth enhancement of the sacrum, means the sugarcane there, and this kind of work can be initiated, which could be a better alternative along with other strategies being followed. Similarly, there is one publication regarding the commercial microbial biostimulants on soil and root, microbial community and sugarcane yield, where this group of workers compared commercially available products, as my friend from Nigeria said that Indian companies are working in his country also for this kind of strategy. So perhaps this study is from somewhere, I don't remember exactly the place, maybe USA or somewhere, where they compared the commercial formulations, and they came to conclusion that 25% reduction in the fertilizers are possible without compromising with yield, without compromising with the quality of crop and its sugar percentage, etc. So this kind of strategy should be followed here, and what should be the possible approaches for the sugarcane crop. I have just tried to jot it down here in this slide, that one should isolate the effective microflora from sugarcane, then selection is there. After selection and after a series of the trials under greenhouse as well as the microplot and small field experiments, one can develop the potential bioinoculant which can be tested at field level. And in our country, since the fertilizer control order is there and biopesticide central insecticide board regulation is there, biostimulant regulation is also there. So after doing all these codal formalities, the specific formulations for sugarcane also can be developed. However, it needs the long-term field evaluation and certain characterization of microorganisms. I always used to say that one should not go directly to the field without properly characterizing those microorganisms. Very casually we used to say that since they are the members from soil, they will not be harmful to the crops. This is true also, but at the same time we have the examples that entire world is faced a major threat and challenge because of only one virus. Our all this plan and everything was at stake 2 years back in the form of COVID. So we should not forget all those kind of threat. If we are manipulating, if we are playing with microorganisms, if we are selectively increasing the population of certain kind of microorganism in the soil system. So just I will take only 2 to 3 minutes. I understand that my time is over, but just to tell you the activities which is being done by my institute, we have two priority, that to conserve the microbial communities involved in global scale, as well as microbial communities might possess intermediate potential uses. We are holding more than 8,000 microbial accessions cultures in our institute. All those are well characterized and characterized for the different potentials in agriculture in the form of biofertilizers, biostimulants and other activities including biopesticides also. We maintain the functional group of microorganisms like more than 200 nitrogen fixers. We have characterized more than 1,500 P solubilizers, more than 400 potassium mobilizers and so on. So we are maintaining a core of microbiota. If you are interested, anybody of the audience, there is a mechanism you can take the microorganisms from our institute, which is the outcome of a well planned research, and you can proceed for the development of formulations. Then we are having the facility for the industries as well as the public sector organizations for the registration of microorganisms. You can register your microorganisms for a soft kind of protection to your research with the institute. We are having association with these public and private sector organizations, and I am open for the collaboration with the industries as well as with the with public sector organizations on various spheres, even sugarcane industries. I am open. I have asked sometimes to my friend also that if there is any plan, any scheme comes from sugarcane industry, we are open for collaboration for development of the solution for some of the things like even the ages old problem of red rot of sugarcane, we can work. We have developed the certain formulations with technology readiness level 1 to 7. Just I will go very quickly. You can see the list from TRL3 to TRL7, several products. Some products are ready for commercialization like BioGrow, BioFast, BioFast Plus, BioZinc, BioPotas, BioSanjeevani, BioCompost, BioShakti and BioShakti 2. So this kind of formulations are ready. We have validated this. I will not take further any further time. Time is running short, but these are the results. If you are interested, we can discuss later on. So I will go very quickly and will with Uttar Pradesh government, we are working with their biofertilizer production units, 10 biofertilizer production unit, and this year we targeted to distribute 17.5 lakh packets of biofertilizer to the farmers. Same thing we want to collaborate with sugarcane industries for the sugarcane farmers. We are working for the Indian soil microbiome project, where we are just conserving all sort of microorganisms, and we are having more than 10,000 formulations, explorations from different countries. So thank you very much. Thank you.
Hello. Myself is Dr. J.R. Kadam, ex-Associate Director of Research, Dry Farming Research in Solapur.
Okay.
Dr. Alok Shrivastava, Director NBAIM UP. You have given a very important information about the conservation, maintenance and utilization of agriculture beneficial microorganisms. But my question is that what is the effect of acetobacter on yield of especially sugarcane? Because VSI is also work on this, right?
Yeah.
At Solapur I have also worked on, and there is a saving of some nitrogen. So what is your vast experience on this acetobacter?
Actually, this is the very prospective microorganisms.
Yes.
Our Sugarcane Institute, Lucknow, VSI, and you told that yourself. You all are working on this. The only thing is that when we are going for commercialization of this kind of formulations, specifically for nitrogen economy, we should consider 2-3 things. Our formulation should be smart enough. Now we are working for the development of smart formulation. That is a strategy. That slide was there, but since time constraint, I did not speak on that. But we are working for synthetic microbials for the agriculture restoration and transformation. What this concept is, that when you are adding any microorganisms including acetobacter in the system, then what it is doing? It is developing its own community in the system. What we call is as a synthetic community in the system. Synthetic microbiota, we are manipulating the natural microbiota of the system. So you should work for the complementarity of this particular bacteria in the system, or you should develop a consortium based on molecular approaches. If not molecular, even the classical approaches, you should mix it with 2-3 other complementary microorganisms which will act as a helper microorganism and support its establishment in that particular environment, particular ecosystem. So this is a very prospective microorganism, and I am sure that in coming time it will be one of the major players for nitrogen economy in sugarcane.
At Solapur, we have conducted a number of trials on use of Acidobacter on sorghum, and we have observed that there is a saving of nitrogen up to 40 to 50%.
Okay, that's good.
Thank you, sir.
Thank you. So you can continue, and if you know the scientific backstopping is required, you can write to us and we will give you some advanced kind of strategy for that.
Sir, you have given a very good lecture on the microbial importance in agriculture. I am Sendil Kumar from NSL Sugars. I am the Vice President.
Okay.
So I have some questions with regard to industrial perspective, with regard to this. So actually, if you see in India, actually in subtropical region, we are having this red rot in sugarcane. And also we have Bukka Boeing.
Yeah.
It is in tropical region. And also OLD, of course, it is a viral one. And also we have a lot of diseases like smut, rust, everything is there. And also with regard to insect pest, we have ESB, early shoot borer, internode borer, top borer, then white grub. These sort of insect pests are also damaging the crop. We want a specific recommendation for each and everything with regard to bio control measures. There is number one. Number two, now it is the period, it is a time for this carbon sequestration, and also we are reducing the carbon emission because of global warming. And also we are getting some carbon credit also, and also it is being marketed. Farmers themselves can do carbon farming, and also they can also have economic benefit out of that. So what is your recommendation with perspective to this microbial participation in sugarcane agriculture?
Okay.
We have so much microbial consortia, and also there should be some specific thing that so that that composting can be done effectively. These are not my questions. I am asking some suggestions on this.
Okay. So thank you, doctor. And actually, I will not use the word recommendation, but what I feel that I can share with you regarding disease management. It's a bit difficult thing in case of sugarcane specifically. Because of certain practices, it's a very long duration crop, and most of the time through irrigation and others, the red rot spread is there. That's why it is difficult to manage. However, some progress has been done by our Lucknow and Coimbatore Institute. The industries are also using trichoderma, effective strain of trichoderma for the management and providing a sort of protection to the crop at the time of sowing. So these strategies are already in practice. The need is that we should develop a system for the early detection of disease. So that part is also very important, that how we are planning the things. I admit that we are poor in that aspect. Not much work has been done. However, some of the institutes are trying to develop a specific kind of biopesticides which can be utilized for the sugarcane. But as of now, the solution is trichoderma for nitrogen economy, acetobacter and some other bacterial formulations are there. But yes, the need is there. Your second question was also related to the management, like the fertilization. For that, we have effective microorganisms, but it is not commercialized yet. So very soon we will be in the position to offer you solution for some of the nutritional requirement of your crop. Regarding waste management, yes, we have the rapid decomposers which is available. If you utilize that, you can considerably reduce the composting time of your waste. There is a package of practices for that, and my institute is working for that. A group of scientists is there. So you can contact, you can write to us, and we can develop that guide. We can provide you some of the effective microorganisms that can fortify the final product after composting, as well as that will shorten the composting time. But in nutshell, since now the sensitization and awareness is there for this particular crop, use of microbiologicals for this particular crop, so I expect some fruitful progress in coming years, maybe 2 to 3 years. Thank you. Just he is asking one question. So 1 minute, ma'am. 1 minute.
Myself,
Yes.
Dr. M.K. Gaur.
Yeah.
I want that, what is the better product developed in the institute? How could it scale up for the industrial purpose, for perspective of that, at that area of the soil health, microbial health? Because the microbial competition will carry on in that place. So how the dose will decide and how it will be beneficial to the industry?
In fact, for that, there are 2 ways. One, our products are available in Agri Innovate site. That is the site of a company, ICR company, who used to commercialize these things through licensing. But directly also, I welcome the industries to give us a letter of intent. In response, we will offer you the collaboration. A collaborative kind of research program is one. Second thing is that we can offer you the license of our technologies, where everything will be given, like what will be the dose, how it has to be applied, and what are the technology behind that. So the companies can just popularize it on their own, or even in collaborative mode with the institute. Thank you very much.